Welcome, medical microbiologists. Today, we're doing a deep dive into the fascinating world of peptide antibiotics.
Oh, those are pretty cool.
We'll be focusing on Chapter 15 of Walsh and Wensvis's book, Antibiotics, Challenges, Mechanisms, Opportunities. We're going to explore how these potent molecules are built and modified, and how they ultimately unleash their power on bacteria.
It's like a microscopic factory churning out these intricate weapons.
Exactly. Ever imagine something like that?
Definitely.
Our goal is to really understand these essential tools in our fight against bacterial infections.
It's vital knowledge for anyone in the field.
So, to kick things off, let's talk about the two main pathways of peptide antibiotic production.
Okay, so we've got the ribosomal pathway.
Right, the one that's probably more familiar to our listeners.
Yeah, it's like protein synthesis 101.
But then there's the non-ribosomal peptide synthetase system.
And RPS for short.
That was definitely a bit more exotic.
Whoa, absolutely. It gets way more interesting.
Let's start with the ribosomal pathway. It's basically like building any other protein, but then things get way more complex with the post-translational modifications.
Yeah, the post-production fine-tuning.
Can you walk us through some of the key players there?
So think of your antimicrobial peptides, like defensins.
Right, the first line of defense.
They're assembled on ribosomes just like any other protein, but then they undergo some pretty serious modifications.
Like what?
Well, for example, they form isulfide bonds, which adds structural rigidity.
Like adding extra armor.
Exactly. It helps them withstand the harsh conditions they encounter when fighting off those bacteria.
Okay, that makes sense. Now, let's talk about the non-ribosomal pathway with NRPS.
This is where things get really wild.
So like nature just threw out the instruction manual and decided to experiment.
Yeah, you could say that.
So unlike the ribosomal pathway, NRPS is RNA independent.
Meaning it doesn't rely on those messenger RNA templates that ribosomes use?
And this freedom allows it to incorporate non-proteinogenic amino acids.
Those unusual building blocks that you wouldn't normally find in regular proteins.
So non-proteinogenic amino acids, that sounds like it would have huge implications for things like drug development.
Oh, it absolutely does.
I mean, imagine the possibilities in terms of chemical diversity.
Exactly. It opens up a whole new world of potential for creating new and effective antibiotics.
Fascinating. So to recap, we have these two distinct pathways. The ribosomal pathway.
The tried and true method with some cool upgrades.
And then we have NRPS.
Mad scientists lab, creating those unique structures.
The book goes into antibiotics as a great example of ribosomally produced peptides that undergo some major transformation.
Antibiotics are really fascinating.
Can you unpack what makes them so special?
Well, it all comes down to lanthionine rings.
Ah, yes, the namesake.
These rings are formed through dehydration and the creation of thioether bonds, which basically link parts of the peptide chain together with sulfur-containing bridges.
So it's not just about the amino acid sequence, but also how they're linked together.
Exactly. And this added rigidity is what gives antibiotics their potent antimicrobial activity.
Makes sense. It's like reinforcing the structure to withstand those harsh environments. The book mentions the discovery of microbisporicin, a particularly intriguing antibiotic.
Ah, yes, microbisporicin.
What makes it stand out from the crowd?
Well, for one, it contains residues like 5-chlorotryptophan and bischidroxyproline.
Those are some pretty unique names.
And they're pretty unusual residues, too.
So it's like nature is constantly evolving, finding new ways to build these molecules.
It's always searching for an edge in the fight against bacteria.
Okay, let's shift gears and talk about another group of peptide antibiotics produced through the non-ribosomal pathway.
Okay, which ones?
The azolyl peptides.
Those are pretty intense.
They sound like they pack a punch.
They do. Thiazolyl peptides like thiostreptin and thiocillins target the 50S ribosomal subunit in bacteria.
And they manage this through a remarkable cascade of post-translational modifications.
It's mind-boggling, really.
Give us the highlights.
Well, you have thiazole ring formation, pyridine ring construction.
It's like watching origami at the molecular level.
Yeah, it's pretty amazing.
But all of this complexity leads to a highly rigidified scaffold.
Which is crucial for their antibiotic activity.
It allows them to precisely target and bind to their site of action.
Like a key fitting into a lock.
And speaking of specific and purposeful modifications, have you heard of polythionamides?
Polythionamides. Oh yeah, they're the kings of post-translational modification.
What makes them so special?
Well, imagine a 48-residue peptide undergoing over 40 modifications.
40 modifications? That's insane.
That's epimerization, C-methylation, even the creation of tert-butyl groups.
Wow, tert-butyl groups. Those are some serious additions. What's the point of all that molecular redecorating?
Well, in the case of polythionamides, it gives them the ability to form pores in bacterial membranes.
So they essentially punch holes in the bacterial defenses.
Exactly. It's a pretty effective way to take down those bacteria.
It's like nature went all out with the weapon design on this one.
For sure.
Okay, we've seen some fantastic examples of how nature modifies ribosomally produced peptides.
Really remarkable stuff.
Now let's dive deeper into the inner workings of those non-ribosomal peptide synthetase factories, or NRPS.
All right, let's get into the nitty-gritty of these molecular assembly lines.
Help us visualize how they operate.
So picture this. Each NRPS is made up of modules, and each module is responsible for adding a specific amino acid to the growing peptide chain.
Like a step-by-step process.
Exactly, just like a factory assembly line.
Very efficient. So what are the essential parts of each module, the key workers on this assembly line?
Well, each module has three main domains: the adenylation domain, the peptidyl carrier protein domain, and the condensation domain.
Let's break those down, starting with the adenylation domain.
Okay, so the adenylation domain, or A domain, is like the quality control manager.
Making sure everything is in order.
It selects and activates the correct amino acid using ATP to charge it up.
Like giving it the energy it needs to do its job.
Exactly.
What happens next?
Well, then the peptidyl carrier protein domain, or PCP domain, comes into play.
What does that one do?
Think of it as a swinging arm that carries the growing peptide chain from one station to the next.
Like a little transporter.
Exactly.
Yeah.
Ensuring a smooth flow along the assembly line.
And then we have the condensation domain.
The C domain.
This is where the peptide bond is formed, right?
Yeah, got it.
Yeah.
The C domain catalyzes the reaction, linking the activated amino acid to the growing chain.
Like a molecular welder, forging those crucial bonds.
A perfect analogy.
The book uses ACV synthetase, which is involved in penicillin biosynthesis, as a simple example.
Oh yeah, ACV synthetase, a classic.
Can you walk us through how that works?
Well, ACV synthetase is nice and straightforward.
Keep it simple.
It's got just three modules. The first one incorporates amino adipate, the second adds cysteine, and the third brings in valine.
And boom, you have your ACV tripeptide.
Just like that.
But the book also mentions vancomycin synthetase as a more complex example.
Oh yeah, vancomycin synthetase is a whole other beast.
Vancomycin is a much larger molecule.
So naturally, its assembly line is a bit more elaborate.
How so?
Well, instead of 3 modules, vancomycin synthetase has 7.
One for each amino acid in the heptapeptide.
And get this, these modules aren't all on a single protein.
They're spread out.
Yeah, they're spread across 3 different proteins that work together.
Wow, that's some serious molecular coordination.
It's truly remarkable how these multi-protein complexes operate.
It's like a perfectly synchronized dance.
A molecular ballet.
So we've seen how these modular NRPS factories can churn out various peptide antibiotics.
Amazing, isn't it?
But one of the big advantages of NRPS is its ability to incorporate those non-proteinogenic amino acids we talked about earlier.
Right, those unusual building blocks.
What's the significance of that?
Well, that's how nature creates those truly unique and diverse structures.
By expanding the structural possibilities.
Exactly. It's like having access to a secret stash of ingredients.
Can you give us some specific examples of these non-proteinogenic amino acids and what they bring to the table?
Sure. So you have 4-hydroxyphenylglycine in vancomycin.
Okay.
And daptomycin utilizes kynurenine.
Interesting.
These unusual amino acids can do all sorts of things like increase rigidity, enhance target binding, or even improve membrane permeability.
They're like adding a special spice to the recipe.
Exactly. They allow nature to fine-tune the properties of these antibiotics in ways that wouldn't be possible with just the standard set of amino acids.
And nature doesn't stop there, right?
Nope. There's one more layer of complexity.
Tailoring enzymes.
The finishing touches.
Think of them as specialized craftsmen that come in after the main assembly line.
They add those final modifications that make all the difference.
What kind of modifications are we talking about?
Well, there are 3 main types.
3.
Glycosylation, acylation, and oxidation.
And each one plays a critical role in refining the antibiotic's structure and function.
Precisely. They're essential for creating those potent weapons that we rely on to combat bacterial infections.
So glycosylation, that's adding sugar molecules, right?
That's right.
It's pretty common in proteins, but what does it do for peptide antibiotics?
Good question. Well, in the case of vancomycin and teicoplanin, the addition of sugars enhances their ability to bind to their target.
And increases their overall activity.
Exactly. It's like adding a sticky handle that helps them grab onto their target more effectively.
So sugars are more than just sweet treats.
They're important players in the fight against bacteria.
What about acylation?
Acylation involves adding fatty acid chains,
Yep.
often at the N-terminus of the peptide.
And what do those fatty acid chains do?
Yeah, they act like anchors, helping the antibiotic attach to bacterial membranes.
Like a hydrophobic tail burrowing into the membrane.
Exactly. It's a crucial modification for antibiotics that disrupt bacterial membranes.
And finally, we have oxidation,
Right, oxidation.
where oxygenases come into play.
Those are some versatile enzymes,
They can do all sorts of things.
like introducing hydroxyl groups, creating cross-links within the peptide, or even forming new rings.
Looks like a molecular toolbox.
And a powerful one at that.
The book mentions vancomycin again as an example of a peptide that undergoes extensive oxidative tailoring.
Vancomycin is a poster child for oxidative modifications.
What's the purpose of all that oxidation?
Think of it like adding extra welds to a steel structure.
Making it stronger and more stable.
Exactly. In vancomycin, those oxidative cross-linking reactions create a rigidified scaffold.
Which is crucial for its ability to bind to its target and inhibit cell wall synthesis in bacteria.
It's a remarkable example of how nature uses oxidation to create a highly effective antibiotic.
So these tailoring enzymes are pretty amazing, aren't they?
Oh, absolutely. They take these already impressive molecules and make them even better.
Adding a whole new level of customization and complexity.
Nature is a brilliant chemist.
That's for sure.
And that brings us to another fascinating class of antibiotics, the lactams.
All lactams, some of the most widely used antibiotics out there.
Absolutely. They're essential in our fight against bacterial infections.
And their biosynthesis involves some seriously elegant enzymatic chemistry, right?
Oh, it's a beautiful dance of molecular transformations.
Lactams are unique because of that four-membered lactam ring.
Right. That's the key to their mechanism of action.
It allows them to target penicillin-binding protein.
Those essential enzymes involved in bacterial cell wall synthesis.
So the book focuses on the biosynthesis of three main classes of lactams: penicillins, cephalosporins, and carbapenems,
Each class has its own unique pathway.
showcasing the incredible diversity of nature's toolbox.
Nature is a master chemist.
Let's start with penicillins and cephalosporins.
Okay.
And they share a common precursor, that linear tripeptide, ACV.
Aminoadipoyl cysteine D-valine.
Right, ACV for short.
And like many of the peptides we've discussed, ACV is assembled by a non-ribosomal peptide synthetase.
The three-module ACV synthetase we talked about earlier.
Exactly.
So far, so good.
But then things get really interesting with the enzymes that create the lactam ring itself.
Okay, this is where the magic happens.
The book highlights isopenicillin N synthase, or IPNS.
IPNS, the key player in penicillin biosynthesis.
IPNS is a remarkable enzyme.
What makes it so special?
Well, it's a non-heme iron oxidase that catalyzes the oxidative cyclization of ACV to form isopenicillin N.
Oxidative cyclization, that sounds complicated.
It's actually pretty elegant.
Can you break it down for us?
Sure. Essentially, IPNS uses oxygen to stitch together the cysteine and valine residues in ACV,
Okay.
forming that critical lactam ring.
So it's like a molecular sewing machine.
A very precise one.
And this process also involves some electron juggling, right?
Oh yeah, it's a 4-electron oxidation with oxygen being reduced all the way to water.
Wow, IPNS is a busy enzyme.
It's got a lot of responsibility.
So what happens next in the pathway? What becomes of isopenicillin N?
Well, for penicillin N, the aminoadipoyl side chain gets epimerized.
Meaning its stereochemistry is flipped.
Exactly.
And this sets the stage for the formation of cephalosporins.
That's right.
Cephalosporins have that 6-membered dihydrothiazine ring fused to the lactam, right?
Right. Unlike penicillins, which have a 5-membered thiazolidine ring,
So how does that transformation happen?
the enzyme responsible is called deacetoxycephalosporin C synthase, or DAOCS.
BAOCS, also known as expandase.
I love that nickname.
Expandase, because it expands the ring.
Exactly. It converts the 5-membered penem ring to the 6-membered cephem ring.
You know, what's fascinating is that it also uses oxygen, even though no oxygen atoms actually end up in the final product.
It's another example of nature's incredible efficiency.
How's that work?
Well, DAOCS is another non-heme iron oxidase,
Okay.
and it uses oxygen along with aketoglutarate to expand that 5-membered ring of penicillin N.
To form the 6-membered ring of deacetoxycephalosporin C.
It's a complex process involving radical reactions and bond rearrangements.
All orchestrated by this single enzyme.
Nature is amazing.
Absolutely. But the diversity of vacolactam biosynthesis doesn't stop there,
Right. We also have carbapenems and clavulinate.
which take a completely different route to forming those lactam rings.
Instead of oxidative cyclization, they use a non-oxidative pathway.
So they don't rely on oxygen for ring closure.
Exactly. They use ATP-dependent lactam syntheses.
So they harness energy from ATP to build the ring.
Nature has evolved multiple solutions to the same problem.
It's a testament to the power of evolution.
It really is.
So in the case of carbapenems, the key enzyme is CARA,
CARA is a fascinating enzyme.
and it works on a different starting material, right? Not ACV,
You're right. CARA works on carboxymethylproline.
a molecule that already has a 5-membered ring.
And the carboxyl group on the side chain and the nitrogen in the ring are perfectly positioned for lactam formation.
So it's all set up for ring closure,
Exactly. CARA uses ATP to activate that carboxyl group.
making it primed for attack by the nitrogen in the ring.
And boom, you have your volactam.
Simple and elegant,
And a similar strategy is used in clavulinate biosynthesis.
but with a different starting material.
Carboxyethylarginine in this case.
Another molecule with a carboxyl group and a nitrogen in just the right spots.
Perfect for lactam formation.
And the enzyme responsible is clavaminate synthase, or CAS.
Another remarkable enzyme in nature's toolbox.
So both CARA and CAS use ATP to power the ring closure.
It's a non-oxidative process.
But the story doesn't end there, right? Even with these non-oxidative pathways, there are still modifications to be made.
You're right, there's always more fine-tuning to be done.
What kind of modifications are we talking about?
Well, both carbapenems and clavulinate undergo further modifications, and many of these involve, surprisingly, oxygenases.
Wait, oxygenases? I thought these pathways were not oxidative.
They are, in terms of how the malactam ring itself is formed,
Okay.
but oxygenases come into play later
To do what?
to introduce additional functional groups or to modify the existing ring system.
So even though they're not involved in the initial ring closure?
They still play an important role in shaping the final structure and activity of the molecule.
Fascinating. What kind of modifications are these oxygenases making?
Well, in the clavulinate pathway, clavaminate synthase is a real multitasker.
It does more than just form the Ball-Tap-Tam ring.
Oh yeah, it also catalyzes hydroxylations, introduces a bridging oxygen atom to form the oxalane ring, and even desaturates the molecule.
That's one busy enzyme.
It's a true molecular Swiss Army knife.
It sounds like these oxygenases are also involved in those crucial stereochemical inversions.
Absolutely, those inversions are essential for the activity of carbapenems and clavulinate.
Because the precise three-dimensional arrangement of atoms is critical for how these molecules interact with their targets.
A slight change in stereochemistry can make all the difference.
So we have these two fundamentally different strategies for building out lactam rings. One that relies on oxidative cyclization, and another that uses those ATP-dependent enzymes.
Two different paths leading to the same destination.
It's a testament to the creativity of evolution.
And it highlights a key theme in natural product biosynthesis. There's often more than one way to achieve a specific function.
Like a master chef versus a skilled home cook.
Both can create a delicious meal, but they might use different techniques and ingredients.
I like that analogy.
Before we move on, there's one more point about lactam biosynthesis I want to emphasize.
Okay, what's that?
We've talked about the different strategies for ring formation,
Right, oxidative versus non-oxidative.
but there are also distinct ways of assembling those precursors.
Okay, this is getting even more interesting.
Remember how we discussed that the ACV tripeptide, the precursor to penicillins and cephalosporins, is assembled on a non-ribosomal peptide synthetase?
Yes, the 3-module ACV synthetase.
Well, the precursors for carbapenems and clavulinate, those carboxyethyl-containing molecules, are assembled in a completely different way.
How so?
They're built from primary metabolite.
Without using NRPS machinery.
Exactly. It's another example of nature's remarkable flexibility.
So no dedicated assembly lines for those precursors.
Nope, they're put together using more general metabolic pathways.
It's like having different manufacturing strategies.
Sometimes you need a specialized factory.
And sometimes a more general purpose workshop will do.
Exactly. And this diversity of assembly strategies adds yet another layer of complexity and wonder to the world of natural product biosynthesis.
Nature never ceases to amaze.
Now, the book briefly mentions a group of antibiotics called hybrid non-ribosomal peptide polypeptide antibiotics.
Hybrid antibiotics. Those sound interesting.
They combine structural elements of both non-ribosomal peptides and polypeptides.
like a fusion of two different worlds.
Exactly. They're like chimeras, blending the best of both worlds.
Chimeras, I like that. So what are some examples of these hybrid antibiotics?
Well, the book mentions streptogrammins.
Streptogrammins, those have two components, right?
That's right, streptogrammin A and streptogrammin B.
Each with its own distinct structure and mechanism of action.
And they work synergistically to inhibit bacterial protein synthesis.
And rifampin, which we discussed in a previous deep dive, is also a hybrid antibiotic.
Good memory. Rifampin has a complex structure that includes a naphthalene ring system from a polyketide pathway and a long chain containing a peptidyl unit.
It's a fascinating example of how nature can mix and match different biosynthetic pathways.
To create these incredibly effective molecules.
These hybrid antibiotics are like nature's ultimate mashups.
Combining different elements to create something completely new and innovative.
But before we get lost in the world of polyketides, let's bring our focus back to peptide antibiotics.
Okay.
And talk about their incredible potential for future drug discovery.
Now this is where things get really exciting.
I'm all ears.
The modular nature of NRPS and the versatility of tailoring enzymes open up so many possibilities for engineering novel antibiotics.
It's like having a molecular toolbox.
Exactly. We can start to design and synthesize molecules with very specific properties.
Tailored to overcome specific challenges.
Like those emerging resistance threats that we're all facing.
The book mentions research on second-generation daptomycin variants.
Daptomycin is a great example.
It's a lipopeptide antibiotic that targets bacterial membranes.
Right. Yes, and scientists are exploring ways to modify its structure to create variants with enhanced activity against those resistant strains.
So it's all about taking what nature has given us and making it even better.
We can learn from nature's designs and use that knowledge to develop new and improved therapies.
We've covered a lot of ground in this deep dive.
We have. It's been quite a journey.
From ribosomal and non-ribosomal synthesis to tailoring enzymes and the intricacies of lactam formation.
It's amazing to think that all of this is happening at the molecular level.
Inside those tiny bacterial cells.
It really highlights the complexity and beauty of nature.
Before we move on to our exam questions, I think it's worth taking a moment to appreciate the elegance and ingenuity of these processes.
I agree. Nature is a brilliant chemist.
Okay, are you ready to put your knowledge to the test? It's time for our deep dive exam questions.
All right, let's put those brains to work.
Get ready, medical microbiologists. It's time to test your knowledge.
See how much you've absorbed from our deep dive into the world of peptide antibiotics.
Here's question number 1. True or false?
Oh, a true or false? I like these.
All peptide antibiotics are synthesized on ribosomes.
That's a tricky one.
Think carefully.
It's designed to make you really think about those two main pathways.
We've been discussing
Ribosomal versus non-ribosomal.
What's the answer?
The answer is, of course, false.
False.
Not all peptide antibiotics are synthesized on ribosomes.
Remember those non-ribosomal peptide synthetases?
NRPS.
Those are the rule breakers.
They don't need ribosomes to do their job.
Nature likes to have options.
Multiple ways to achieve the same goal.
Exactly. Ribosomes are just one tool in nature's vast toolbox.
All right, ready for question number 2?
Bring it on.
What is the key structural feature that gives antibiotics their name?
The antibiotics, those fascinating molecules with all those post-translational modifications.
Think back to our discussion.
What sets them apart?
The answer is lanthionine rings.
Lanthionine rings, of course.
Those unique rings formed through dehydration and the creation of thioether bonds.
They're the hallmark of antibiotics.
Adding rigidity to the peptide structure and boosting their antimicrobial power.
A perfect example of how post-translational modifications can completely transform a peptide.
From a simple chain of amino acids to a highly specialized weapon.
Okay, question number 3.
This one's a bit more challenging.
Name 3 types of post-translational modifications commonly observed in peptide antibiotics.
3 types.
Think about those tailoring enzymes,
The molecular craftsman.
adding the finishing touches.
What tools do they use?
The answer is glycosylation, acylation, and oxidation.
Those are the big 3.
Each one with its own unique impact on the antibiotic's properties.
From target binding to membrane localization to overall stability.
Nature has a knack for fine-tuning these molecular weapons.
Remember those examples we discussed? Glycosylation in vancomycin, acylation in lipopeptides, and the extensive oxidative tailoring of vancomycin.
Nature's arsenal of tricks is truly impressive.
Ready for question number 4?
Hit me with it.
Describe the role of isopenicillin and synthase, or IPNS, in volactam biosynthesis.
IPNS. That brings us back to the chemistry of lactam ring formation.
It's a crucial step in the creation of these essential antibiotics.
So what does IPNS do?
IPNS is responsible for the oxidative cyclization of ACV
That linear tripeptide.
to form isopenicillin N.
And IPNS is a non-ham iron oxidase, right?
Exactly. It uses oxygen to create that phylactam ring.
A beautifully orchestrated molecular dance.
Okay, last question.
The grand finale.
What is the key difference in phylactam ring formation between the penicillin cephalosporin pathways and the carbapinamidoclavulinate pathways?
This one really tests your understanding of lactam biosynthesis.
Think about those different strategies.
Oxidative versus non-oxidative.
What sets them apart?
The answer lies in how the lactam ring is formed.
Whether it's through an oxidative or a non-oxidative process.
Penicillins and cephalosporins rely on oxidative cyclization using enzymes like IPNS and DAOCS.
But carbapinams and clavulinate take a different route using ATP-dependent malactam syntheses.
They harness energy from ATP to build the ring without needing oxygen.
Two different paths, same destination.
Nature's ingenuity at its finest.
That wraps up our exam questions. How did you do?
We hope you enjoyed testing your microbiology knowledge.
And that you gained a deeper appreciation for the intricate world of peptide antibiotics.
From those basic building blocks to those remarkable enzymatic transformations.
These molecules are truly amazing.
But before we say goodbye,
We want to leave you with a final thought.
we've seen how nature crafts these molecules with such precision.
The elegance and ingenuity of it all.
It's inspiring. But what if we could take what we've learned and apply it ourselves?
Could we design and build our own peptide antibiotics?
Tailor-made to combat those emerging resistance threats,
It's a challenge for sure.
but also an incredible opportunity.
To push the boundaries of drug discovery.
We encourage you to explore Chapter 15 of Walsh and Vencevich's book
Antibiotics. Challenges, mechanisms, opportunities.
and dive deeper into the research in this field.
There's so much more to discover.
And who knows, maybe you'll be the one to create the next generation of life-saving antibiotics.
And to leave you on an inspiring note, here's a short poem.
A poetic ode to these molecular warriors.
From simple chains to rings so tight, peptide antibiotics wage a valiant fight. Nature's arsenal, a wondrous sight, evolved to conquer with all their might. From ribosomes or crafted anew, a symphony of structures strong and true. They bind and block and pierce right through, protecting life, a vital crew.
Until next time, keep exploring, keep learning, and keep diving deep into the fascinating world of science.